what is the research which is going on in entire world and India, and finally in Goa regarding this termitomyces. I'm going to give a brief journey. I have already understood through videos what I have Dr. Nandkumar have screened for y'all, that what is the importance of this association of termites and termitomyces, that is Roinichi. Now what does the termites do is that they get all the surrounding litter, plant litter which is available, and they create an underground, that fungus comb which is there in the termite mound, which is normally called as locally known as olmo, and through that these termitomyces grow. Now how does they benefit each other? Now termitomyces, they help to digest this plant litter which is present in the termite mounds and give food for termites. So besides that, termitomyces are in a form of small white globules inside the mound, fungus comb. So another way they benefit is that termites get to eat this and get a nitrogen source, because this fungus comb lacks nitrogen, or it is in a minimal amount, and only carbon content is very high because litter is rich in carbon. So this is a mutual association which they have, and that's why they need both the partners for their survival. Now what is the trend which is taking place in the research? Now previously in 1990s there were research, you can see there was very less number of publications, but as from the 2000 onwards, you can see there is high demand for this genus, and there's a lot of work which has been carried out. So it shows that the bar had increased to more than 4,000 publications. So basically what scientists have been working regarding this termitomyces genus is that mostly it's like ecological understanding. What is the role of this termitomyces and termites in the ecosystem? How they benefits, because there is a lot of litter conversion studies which have been taking place, nutritional, additional, and a lot of other studies also. And the other part is diversity and taxonomy. What we see in the market, you may just say it is Roinichi, but scientists, they sit and try to see the morphological changes in each and every fruit body, what you see. So basically then they identify and screen and give a different names. So there are species names. So in that way, some are totally specialists in only understanding the diversity. And that is the study called as taxonomy. Now further on, now people have gone more advanced by using a molecular techniques. Now what is a molecular techniques, wherein they use different probes and accurately identify this species, like how we have barcoding. So based on that, now phylogenetic studies have been also been carried out, wherein you can find the evolutionary scale. How old was this termitomyces originally occurring, or how old is a termite relationship? So that is studied by molecular studies and phylogeny. Now also there are scientists which are working in especially only in the nutritional areas. Now what is why it is important to the nutritional? Because now you know through the ethnomycological knowledge that these mushrooms are edible, and besides that there is also like they are used in medicinal purposes by local people earlier, by tribal people. So this has developed an interest in scientists to know what exactly is there in these termitomyces and identify such, those compounds and record. Now the nutritions, like they have been reporting like protein content, carbohydrates, fatty acids, or other novel compounds which have been only specifically found in termitomyces. Then there are other advanced studies have been also carried out regarding the various bioprocesses, wherein a process has been identified and to give a final product, which is a bioproduct. So that is detailed studies which have been studied. Now basically what are the materials which they use for their study? Now the entire study is not just based on the fruit body, but there are all different stages which you can see through the entire section of the termites. So it starts with termite gut contents onwards, because we know that this anamorphic stage of termitomyces has been consumed by termites, and again they redeposit into the fungus comb, and thus the journey continues. So the research starts from there in termitomyces. Then it goes to the fungus comb, wherein the fungus comb, which we have seen there is anamorphic stage with white nodules, and it also has the ramified mycelia in it. So that is also been used as a source material. Then the sporodocia, which is the white nodule, which you can see, it is a zoomed magnification. And then different types of fruit bodies, which are all termitomyces, but they appear in different forms. Then there are basidiospores cultures you can obtain. Now these is the spores which are given by this entire cap of the fruit body. So each single spore is a seed. So they can give rise to a single colony or a new fruit bodies. Then there are sporodocia. From this also cultures are obtained by the scientists and also from the fruit bodies and are further worked on. Now this is a global feature of the diversity which is there in entire world, and you can see that these are the several countries which have reported termitomyces in their country, and this is a diversity. Like for example, in India we have around 28 distinct species, and overall in the Index Fungorum there is one of the fungal databases which reports there are 90 distinct species of termitomyces, which is a huge diversity, and from that we have around 31% in India, followed by the Chinese, which has around 28% in China, and so on. So now what is this? I have break down the whole research, the global research, into the different country-wise, so that you can get an easy prospect what each country is working on. Now mostly the European countries which are there, that is like Denmark, Netherlands and all, they don't see the fruit bodies directly, but they have these termite mounds and the anamorphic stage. So most of the people are working with that as a source material, and they have studied the mostly phylogeny, evolutionary studies have been carried out. Further, China has had using a lot of advancement in the technologies, and they have reported novel compounds, cerebrosides, which are along with the Japanese, and they have identified these are called as termitosesphines and termitosemides, which are one of the very interesting compounds which have been documented from this genus. Then Malaysian people have also been worked on and identified important proteins which play a role in future medicines and other immunostimulatory studies. So these are the compounds which you can see which are only found in termitomyces, and they are known in future we can use as a drug source for curing different type of neuronal diseases. So we need to save this species, so, and we must not lose this germplasm just because of our taste. So this is very important for us. Secondly, there are many other African countries and other countries which have been basically working on the nutritional aspects and other even fructification has been attempted, like producing fruit bodies in vitro, but it ever not successful, as Dr. Nandkumar said. Now what basically Indians are working on? We have very huge diversity of 31%. So most of the people, there are taxonomists, ecologists who have been working, and ethnomycological knowledge has been also documented. So this shows there is a lot of transition of data from generation to generation. Until date we have this diversity. So it's a very high time that we need to preserve it. Now besides that, Indians have extensively worked on two different areas, that is enzymes and polysaccharides. Now enzymes, you can see there is a list of enzymes which have been obtained from the single genus. So you can see there is a lot of potential in industrial scale also. And this can lead to a lot of job opportunities and new discoveries of more important products which can be done, or like we can use all these enzymes and produce like how normally now there are many lactases which are being commercially used from the other sources. So future we can use termitomyces as a source. So there are also polysaccharides which have been reported. Besides, this is just a view I wanted to give which are the different institutes in India working on. There are many institutes which are working in a collaboration, but actively working groups are very few. So you can see that there are top institutes which have been working. That one of them is the Indian Institute of Chemical Biology, which is a CSIR institute, which is extensively working on this genus, and there's all these enzymes and all have been documented by them. Then secondly, we have Goa University, which have been actively working from past so many years, and you have seen and witnessed the presence of entire research journey. And then other universities which are present in Mangalore and West Bengal and Punjab universities have been worked on. So in Goa, you have already seen the research work, and as you know, Dr. Nandkumar Kamath is the pioneer in this area in Goa, and we're working on this genus for so many 35 years. Now, you can see the different videos what he had uploaded on the YouTube for educational purposes. So I request you all please go and check out, because every video you get a lot of knowledge and you will have a feeling that you need to conserve this genus. Secondly, there's a different photographs also which have been uploaded. You can see that. He always writes in a newspaper. Public social awareness has been carried out by Dr. Nandkumar Kamath, and he writes in the columns. So this is this shows that how much concern he has towards saving this genus, and I hope that each and every one of you all will take part in this mission of saving Termitomyces. You can see also the other articles which have been published. So what I have been working on in this genus area, I've also tried to record the diversity of which is present in Goa for past 7 years, and now we have seen that there is high demand for edible protein. So why not try this in other alternative ways in growing this mushroom? So we are trying to work on getting this Termitomyces in the cultural form in the laboratory, and then trying to grow it in the liquid form, and getting the entire biomass in the liquid submerged laboratory conditions, and thus we can give a future mycoprotein. Secondly, we have also seen that fruit bodies produce a very important compound, that is black pigment, and which is called as melanin. So that has also been characterized. So basically, we start from the identification process of the mushroom at the laboratory scale, wherein we use different keys and publications, texts, wherein they describe the species description. We match them not just by the morphological observance, but by we do micromorphology of these various fruit bodies and all, and we try to identify the differences across each species, and then accordingly we give them the name, and then we isolate the pure culture of each species by using the different methods, and this is how they look in the culture form. Now these are same Termitomyces what you eat, but now that is in the field and this is in the lab. So they grow exactly in the two-dimensional form and the mycelial culture. So you can see that in the world culture collections there is around 15 species which have been documented and deposited, and by different database, different countries, that is Africa, Taiwan, India, Thailand and Philippines. From this, 6 species have been deposited by Goa, that is Goa University Fungus Culture Collection Unit. So further on, those same cultures, we study them for the micromorphology by using different microscopes, that is optical microscopes or scanning electron microscope, to get a more higher resolution details and to identify the key points and the distinct features across each species. So this is what we have done, that we have first time seen the morphological differences which are taking place across the different species of Termitomyces. Globally, before the researchers have not described the morphology of these Termitomyces in the liquid cultures, but basically some of the researchers have been working on the submerged or stationary cultures, but they have not exactly screened for different morphologies and to identify them based on their morphology in the shaken conditions or liquid conditions. So we in Goa, we have given the different names for the shapes as per the literature which have been applied to the different other mushrooms. So we have seen that the different morphological variations like spiky pallets, aggregates, fluffy or compact pallets. So these are the different morphologies of Termitomyces in the shaken conditions. Now, similarly, we have also applied the scanning electron microscopy to get a better view of the same biomass which we obtained in the shaken conditions, which are called as pellets. And this work has been published in the International Journal of Biotechnica and Histochem, wherein this entire research work is available for the scientific community. Now, FTIR spectroscopy is another technique which we apply to screen the different changes which are taking place in the biomass and record the variation across the species again. And based on that, we were able to screen or select the best strain for the which can be used for the industrial purpose. So we have used different techniques, like from the optical microscope to scanning electron to the FTIR or other form of techniques to get the ideal strain which can be used for the industrial purpose, which need a lot of further work which we need to carry out to get an ideal protein source which can be marketed in the future, such as mycoprotein. So this work has been also published and made available to the scientific community. And another part which we have been worked, as I said, it's a pigment which we have isolated, which is sulfur-rich pigment from the Spermatomyces fruit body. And this is a black pigment which is commonly found, which is there in all the universe. What you see like black, for example, hair, we have melanin in it, but only the structure is slightly different, or the composition is different, but the role is same. So that work which saw that is accepted in the International Journal of Mycology. So this is a wide application which we have of this pigments, and we can use it in the future for industrial purpose. As we know, it can play a major role in drug delivery, then the cancer treatment therapies. And these are the important things because these are upcoming problems which are there. So we need to solve them by using the edible source of melanin, which is the melanin is edible. We have to get it from the edible mushroom, so it will have minimal effect. Okay, we don't have any side effects because it comes from the edible source. We also added a new record for India, that is Spermatomyces vulgarizers, which was found in Goa. So that has also been published in the Kavaka journal. Besides that, I have also presented my papers for the international conferences, and I made the data available for the scientific community. So thank you.